All right, so today we're going to start talking about the different phases of mitosis. We learned in the cell cycle unit that mitosis is how the process that cells go through to divide. And before we start talking about mitosis, I want to go through a couple of vocab words with you guys that will help you. You're going to hear it when we are learning about mitosis. You're going to hear it when we're talking about genetics in the next upcoming units that we're going to start. So let's get started with this. So what I want you to know is the difference between a chromosome, chromatin, and chromatid. So a chromatin, let's start with that. So as you can see in the pictures here, the chromatin is it's this unwound form of DNA that exists in the nucleus, and this is the form that usually exists when the cell is in interphase, just doing its thing before the DNA is going to copy. It's unwound because it just makes it easier for the gene and protein synthesis to happen when the DNA is not all tightly wound up. But when a cell is going to go through cell division, it needs to wind up that DNA really nice and tight into a chromosome form because your DNA is really long. And if you've got a double set in there because it's copying the DNA in order to divide, there's going to be so much DNA in there that it's got to get in this tighter form. So the chromatin is this loose form that exists in the nucleus before the cell is going to divide. The chromosome is this tightly wound form that is kind of more that shape that you typically see in the textbook and that type of thing. It's the shape that the chromosomes take before they're going to divide. And then the last thing you need to know is the word chromatid, and that is as we learned in the cell cycle, the DNA copies itself. It makes a copy of itself right before its cell division, and it does that so that when the parent cell becomes two daughter cells, each daughter cell will have a full set of DNA. The DNA will split into two, and we're going to see how that actually happens during the phases of mitosis. But for now, just know that the DNA has to copy itself, and those copies have to find each other before cell division can happen and the DNA can be split into two new cells. So the two forms will find each other, their exact pairs if all the copying mechanisms worked, and they're going to line up in the center and they're going to join at what's called a centromere, is this centerpiece that holds them together. Again, you're going to see this as we're learning about DNA and mitosis, but I want you to know these things to start off. Okay, so let's start looking at how mitosis really works. So just to give you a definition, a formal definition of mitosis, we're going to say it's a continuous process. So once it starts, it moves forward in a continuous process. However, biologists have divided the mitotic phase into four main stages. You have prophase, metaphase, anaphase, and telophase, and they happen in that order. Prophase is going to be the first phase, and it's the longest phase, in which those chromatin bundles, those loose bundles of chromatin inside the nucleus, the chromatin bundle up to form chromosomes in the nucleus. Each chromosome will consist of a pair of sister chromatids that are joined at the centromere. So remember, the DNA copy has already been formed, so these chromatin are going to bundle up to become tight chromosomes, and they're going to find their sister chromatid pair, and they're going to join up in the center at centromeres. The other thing that happens is the nucleolus disappears, and the cells will stop making ribosomes, and a football-shaped structure called the mitotic spindle is going to form, and these mitotic spindles will start tugging the chromosomes toward the center of the cell. So after prophase has happened and those sister chromatids have found each other and they're joined by the centromere, we're going to get to the second phase of mitosis. It's a brief phase, and it's called metaphase. And this is where all the chromosomes are then going to line up in the center of the cell. So you've got your cell, you've got your chromatids joined together. They're right at the center of the cell. And then the next phase of anaphase is going to happen. So this is the third stage where your sister chromatids separate, and each chromatid is now considered a daughter. And the way that they separate is you have those chromosomes lined up in the center with those spindle fibers attached to them. And those spindle fibers are going to start to shorten. And as they shorten, so let's say these are your two sister chromosomes and they've got the spindle fibers attached to each one of them. As they shorten, the spindle fibers shorten and they pull on the chromosomes, and the chromosomes are going to start to separate and move to the other ends of the pole of the cell. All right, so once those sister chromatids are starting to separate and move to the opposite ends of the cell, we enter the final stage of mitosis, which is called telophase. And this is where those chromatids are actually brought to the opposite sides of the cell, and two new nuclei are formed around those sets of chromosomes. And there's going to be one on each side of the cell. The two daughter cells are formed when a new double membrane forms, allowing the cytoplasm to divide and separate by cytokinesis. So telophase is the phase where the chromosomes have moved to the opposite ends, a new nucleus forms around the chromosome, and then cytokinesis begins and the cell split into two.